Now, when bacteria move, they're responding to their environment, and that response can come in a number of ways. It can be by chemotaxis, in which they're responding to molecules in the environment, either that are attracting them or repelling them. That can work either way. You can have phototaxis, where a microbe maybe is moving towards light, or aerotaxis, where it's moving towards oxygen or away from oxygen, depending on the microbe's need. When bacteria move, they move in what we call a run and tumble fashion. So basically, the microbe will beat its flagella in a, or I should say rotate its flagella in a counterclockwise fashion that will cause it to run. Okay, so it'll move in a straight line in one direction. What's causing it to run? Well, maybe chemotaxis. Maybe there's some sugar over here, so it's moving in that direction. So it'll run in that direction for a while, but then it'll tumble, where it'll stop, reverse the direction of the flagella. So instead of going counterclockwise, they started out going clockwise, and this causes the microbe now to tumble. It'll come out of that tumble, and it'll run again. And then it'll go into a tumble. It'll come out of that tumble, and it'll run again. And how it comes out of the tumble is kind of random. It just comes out of it and then goes. Ideally, if you're heading in a direction with long runs, you'll eventually get there. What if you don't want to go away from where you're at? What if you wanted to stay here? Well, the microbe employs a different strategy. It reduces the size of its runs so that it runs for a little bit, then it'll tumble, come out of that tumble, run again, tumble, come out of that tumble, run again, and because it reduces its runs, it has a tendency to keep it in a general vicinity so that it doesn't move out of this area. So if the sugar was good here and it didn't want to stay, it would increase its frequency of tumbling, reduce its run, so that it can stay in that area. But if something is attracting it away from that area, then it's going to have long runs, and its frequencies are going to be, or its tumbles are going to be less frequent. Any questions about this notion of bacterial movement? Yeah.
So the further the attraction, the more it'll continue to run and tumble?
Yes. Well, see, you have to imagine that there's probably going to be a gradient. Say we're talking about glucose. Okay, so let's say that, I'll use pink here. There's 0 glucose here, 0% glucose here, and 100% glucose here. So there's probably a gradient that's going to continue to attract it in that direction. So if it tumbles here and then, say, shoots off this way, it'll tumble again and hopefully end up going back this way. But as long as the attractant is still pulling it in one direction, its runs are going to be longer and its tumbles are going to be shorter. Now imagine if it starts off this way here, then the tumbles are going to probably shorten down so that it can head back the other direction. One more thing that we'll say about the, I got some time here, the structure of the cell with regards to external stuff outside the membrane. They also have what are called pili. Pili are these extensions, shorter, thinner than flagella, but they serve a number of purposes. They can serve as an attachment site, either to a surface or, in a lot of cases, it's the end of these pili that are adhering to surface receptors on cells that then allow them to infect those cells. So a lot of times attachment is taking place here at the pili. Anyway, attachment can happen, movement, they can move across the surface kind of like a twitching thing. And then conjugation. Pili are involved in conjugation. Conjugation is a process by which the bacteria essentially have sex. They come together and this microbe passes its genetic material over to this microbe, essentially exchanging genetic material. That's as close as bacteria get to having sex, and we'll say more about that shortly here. What do we find inside the cell? Of course, you're going to have the nucleus, excuse me, take that back, not the nucleus, the genetic material, and you'll see it referred to as a nucleoid, just because a lot of times it's located in one general area. But the point is that it's not a nucleus because it's not bound by a membrane. And that's one of the distinguishing characteristics between prokaryotes and eukaryotes. And it's circular, it's a singular genome, double-stranded DNA, circular molecule. Bacteria also have what are known as plasmids. Plasmids are little additional pieces of genetic material, often picked up from other microbes through the process of conjugation. And these plasmids can impart to the microbe a whole host of new characteristics, maybe new proteins that they can use for digesting food, or new secretions, new toxins that they might be able to release. We'll also find ribosomes. These ribosomes are different than eukaryotic ribosomes, different than the ribosomes we have. They're slightly smaller. This is where protein synthesis is taking place. There may be storage vacuoles. I think that's enough put on there. Sometimes you can find gas vesicles there as well. And your book talks about endospore formation. Some cells will form an endospore. But I think that'll cover it for now. So that's the structure of a prokaryotic cell. You should understand or know all of those parts. This is a great essay question. I could ask you to draw this structure and then identify the parts, maybe give me a function for each of the parts or what a role might be for each of them.
So know the cell structure?
Mm-hmm, absolutely.
Okay.
Okay, so I'll call you, come down and get your exam. I'll try to meet you where I can. Augustin Castro. Augustin. Mario. Yeah,